So the title of my talk is Extracellular RNA, a trigger of sterile inflammation, which I want to show you. And that this extracellular RNA initiates natural bypass growth, what we call arteriogenesis. So I'm from Munich in Germany. I'm from the Walter Deindl Center of Experimental Medicine, from the Ludwig-Maximilians-University there. So, arteriogenesis. Arteriogenesis is the growth of a natural bypass. So what you can see here is a human heart. It's an angiogram of the human heart with a completely occluded coronary artery. So I think everyone expects that this patient suffered from a myocardial infarction. However, he did not. He was brought into the clinic and diagnosed with a stomach CA. And that he has a completely occluded coronary artery was just found by chance. So neither he nor his physicians had any idea that he has a problem with the heart. So what happened? The process of stenosis here occurred very, very slowly. So it was not acute, but very, very slowly. And the blood, which normally flows through this coronary artery, was redirected in one of those pre-existing collateral arteries. And due to the increased mechanical load, due to the increased shear stress, this collateral artery started to grow and finally created a natural bypass, what you can see here, which completely overtook the function of this occluded coronary artery. And this natural bypass always appears in this typical corkscrew formation. So what happens is that due to the increased mechanical load, the endothelium gets activated, leukocytes, in particular monocytes, are recruited, which supply a variety of growth factors and cytokines to the growing vessel, finally resulting in vascular cell proliferation and vessel growth. Though this whole process relies on an inflammatory reaction and presents a sterile inflammation. So, from a previous study, which we have published in Cell Reports in 2016, we know a little bit more about the molecular mechanisms, how the story works. So there we show that due to the increased shear stress, the platelet receptor GP1B alpha gets activated. Thereupon, platelets interact with neutrophils, and this PNA, platelet-neutrophil aggregates, are formed. Concomitantly, neutrophils are activated. There is an extravasation of these neutrophils, and these neutrophils produce via the NOx2 reactive oxygen species, so ROS. And this ROS activates mast cells. Then these mast cells release some growth factors. But what they also do, and this is very important, they increase the bioavailability of the inflammatory factors, TNF-alpha and MCP1, which on the one hand in a feedback loop results again in recruitment of neutrophils and activation of mast cells, and on the other hand on the recruitment of monocytes, which mature to macrophages in the perivascular space, and there they apply all the growth factors and cytokines to the growing vessel. Now, of course, one question came up, and this was, how can shear stress activate platelets? So how is the platelet receptor GP1B alpha activated by shear stress? And this is where RNA, and in particular extracellular RNA, comes into play. So all of you have learned, I think in school, at least I have learned at school, that RNA is inside a cell. But RNA is also outside of cells. And this has already been found in 1947. So in the plasma of healthy individuals, RNA proteolipid complexes have been found. So about 60 years later, Kannemeyer et al. Have published that extracellular RNA is a cofactor in thrombus formation. And Sylvia Fischer has published in 2007 that extracellular RNA also increases vascular permeability and promotes edema formation. And we have found in 2012 that extracellular RNA promotes leukocyte recruitment. So for that study, we used a murine cremaster model of inflammation, and there we could show that when we treat the mice with RNA, with extracellular RNA, which we isolated from another mice, we find increased numbers of mononuclear cells, and extracellular RNA was as inflammatory as TNF-alpha, which was our positive control. So, since extracellular RNA is able to recruit leukocytes, we thought whether there's a function of extracellular RNA in arteriogenesis, which presents a sterile inflammation, which is an inflammatory process. So to investigate whether RNA might play a role, we first of all looked whether RNA is released from endothelial cells due to shear stress, since shear stress is a driving force of arteriogenesis or for arteriogenesis. So first of all, we performed some in vitro studies and applied shear stress to bovine aortic endothelial cells, and indeed found that shear stress of about 12 dyne, we used several dynes, but also 12 dyne, indeed results in increased levels of RNA, of extracellular RNA in the supernatant of those cells. The level of LDH was not increased. LDH is a marker for cell damage, so this was not changed compared to the static situation. So this clearly indicates that the extracellular RNA from the endothelial cells is not released due to cell damage, but actively. So then we were, of course, interested, what about extracellular RNA under conditions of collateral artery growth? And therefore, we used our mouse model of peripheral arteriogenesis. So what we are doing here is a mouse, of course, the two hind limbs. On the left leg, this is the left leg of the mouse, here you can see the femoral artery, and here the pre-existing collateral arteries. On the left leg, we do a sham operation, and on the right leg, we ligate the femoral artery, so we do an occlusion. So due to occlusion of the femoral artery, the blood flow is redirected in those pre-existing collateral arteries, and they start to grow and appear in this typical corkscrew formation. Yeah, you can see it also on these superficial pictures where we have infused latex. Here the pre-existing collaterals, and after ligation of the femoral artery, when the blood flow is redirected, these collateral arteries grow and appear in the corkscrew formation and finally overtakes the function of the occluded artery. Now, using this animal model, we looked whether in these collateral arteries, due to shear stress, RNA might be released from endothelial cells. So what did we do? We ligated the femoral artery, and in parallel, we administered an antibody against rRNA, ribosomal RNA. And this antibody was labeled with a green fluorescent dye, and this antibodies cannot enter a cell. If we get a signal, a green signal, in our muscle tissue, in our tissue samples, this must be extracellular RNA. And indeed, we found when we ligate the femoral artery that in growing collaterals, there is extracellular RNA at the abluminal side of endothelial cells. The endothelial cells are marked with CD31. So at the abluminal side of endothelial cells, we find extracellular RNA. only in growing collaterals, however, not in resting collaterals. This is the sham-operated side of the mouse. Here also is a pre-existing collateral, and here you can see more or less no extracellular RNA. Now, we were asking about the relevance of this extracellular RNA, and we measured reperfusion recovery. So if we ligate, or let's say the other way around, so before ligation of the femoral artery, the perfusion of the pons is about 100%. So on the right leg and on the left leg. So 100% divided by 100% is 0. But after ligation of the femoral arteries, the perfusion drops to about 10%. But with the time, the perfusion increases again, and this is due to collateral artery growth. So if we treat those mice with RNase, we could see that the perfusion recovery was significantly reduced, whereas the treatment with DNase has no effect. So RNA degraded extracellular RNA and perfusion, the perfusion recovery was worse. Digestion of extracellular DNA had no effect, so it must be somehow RNA. And then we did the same experiment and administered not RNase A, which is from bovine, but we administered recombinant active human RNase 1 and also inactive human RNase 1. So with this experiment, we wanted to investigate whether the reduced perfusion recovery we have observed here is due to RNA digestion or to a yet unknown function of RNase. And indeed, we found that the administration of active RNase resulted in reduced perfusion recovery, whereas the treatment with inactive RNase shows no effect. And these results were confirmed when we measured the inner luminal diameter of growing collaterals after 7 days. So the treatment with RNase as well as active human RNase 1 significantly interfered with collateral artery growth and resulted in a reduced inner luminal diameter compared to saline treated mice, as you can see here. So, as I showed you before, arteriogenesis is dependent on the recruitment of monocytes because they supply all those growth factors. And again, we found when we treat mice with RNase after induction of arteriogenesis, we found reduced levels of leukocytes in the perivascular space of growing collateral arteries. So here we investigated CD45, which is a panleukocyte marker. And at day 3, we found indeed reduced numbers of monocytes. Here the markers F480. The number of leukocytes in the blood was not reduced, clearly showing that the reduced accumulation of leukocytes in the perivascular space was due to reduced extravasation and not due to a reduction in the release of leukocytes from the bone marrow. So for the recruitment of monocytes, we need mast cells, and these mast cells need to be activated by neutrophils. And we also investigated mast cell degranulation and neutrophil recruitment in mice treated with RNase A, respectively with human active and inactive RNase 1. And again, we found, and this are now results from day 1 after induction of arteriogenesis, that we have reduced numbers of perivascular leukocytes, and at day 1 we find significantly reduced numbers of neutrophils in RNase A and RNase 1 treated mice. And the reduced numbers of neutrophils is associated with a reduced number of degranulated mast cells in RNase A and RNase 1 treated mice. And these mast cells, of course, the activated mast cells we need for monocyte recruitment. So now, for activation of neutrophils, I explained to you that we need this platelet-neutrophil aggregate formation. And here we found that we treat the mice with RNase, we have reduced levels of PNA aggregates. Now, these platelets need to be activated, first of all, to form PNAs via the platelet receptor GP1B alpha. And it's well described that one major ligand for the platelet receptor GP1B alpha is von Willebrand factor. So we look for von Willebrand factor knockout mice and induced arteriogenesis in this knockout mice. And indeed, we found that von Willebrand factor knockout mice also show reduced platelet-neutrophil aggregates. And when you look in the literature, you find that von Willebrand factor is released from viable Pallade bodies from endothelial cells upon activation of the VEGF receptor 2. So therefore, we blocked the VEGF receptor 2 with semaxanib, and again found reduced numbers of platelet-neutrophil aggregates. And that's this reduction of platelet-neutrophil aggregate formation is not due to a reduced number of platelets or neutrophils. We investigated the number of platelets and neutrophils in the peripheral blood, and none of those treatments resulted in reduced numbers of platelets or neutrophils in the blood. So of course now we were interested whether RNase treatment itself interferes with von Willebrand factor release. And therefore again, we induced arteriogenesis by femoral artery ligation. And this time we administered an antibody which recognizes von Willebrand factor. This antibody was also labeled with a fluorescent dye, with a green fluorescent dye. And indeed, we found that ligation of the femoral artery, so induction of collateral artery growth, results in the release of the von Willebrand factor in growing collaterals. This was not found in resting collaterals of sham-operated animals. However, if we treat the mice with RNase A, we do not find any more the von Willebrand factor release from these growing collaterals. The same was true when the mice were treated either with semaxanib or with DC101, which is a blocking antibody against VEGF receptor 2. So also blocking VEGF receptor 2 interfered with the release of the von Willebrand factor from growing collaterals. Now, it is well described, and this was already published in 2005, that to translate shear stress into all these biological and biochemical reactions, we need a mechanosensory complex, and this consists of PCAM1, VE caterin, and the VEGF receptor 2. And we also know that this receptor must be activated by VEGF-A, and we know that the endogenous levels of VEGF-A are sufficient to activate the VEGF receptor. They are necessary and sufficient. So if you exogenously administrate VEGF-A, you cannot further promote arteriogenesis. So therefore the question arises how the VEGF receptor is locally activated in growing collaterals. And interestingly, it has been published that enhanced VEGF signaling is mediated by VEGF if VEGF is able to act the VEGF receptor 2 together with its co-receptor neuropilin-1. So VEGF has to bind to both of the receptors to locally activate the VEGF receptor in an enhanced way. And we have previously shown that extracellular RNA mediates the interaction of VEGF-A with this both receptors, so with VEGF receptor 2 and NRP1. So, so therefore, we tested what about activation of the VEGF receptor? So if the extracellular RNA mediated effects on leukocyte recruitment are indeed mediated by the VEGF receptor 2. And here we again used our murine model of our murine inflammation model, so the cremaster model. We again administered RNA and found that RNA, of course, results in increased adherent leukocytes in vessels. So what you can see here, finally, this is a movie, and it even works stronger in that case than TNF-alpha, which was the positive control. However, if we concomitantly block the VEGF receptor with semaxanib, as shown here, the effect of RNA to recruit leukocytes is gone. So from all this data, we conclude that upon shear stress, RNA is released from endothelial cells, and this RNA mediates the interaction of VEGF-A with VEGF receptor 2 and its co-receptor neuropilin-1. This results in the release of the von Willebrand factor, which then activates platelets via the GP1B alpha receptor. This induces a PNA formation, platelet-neutrophil formation. Neutrophils are activated. And as already shown before, so we are here, shear stress, extracellular RNA is released, VEGF receptor 2 is activated, von Willebrand factor is released, platelets are activated, PNA formation, then neutrophils, we have the extravasation, ROS is produced, which activates mast cells, then we have an inflammatory environment, if I have increased levels of MCP1, TNF-alpha. In a positive feedback loop, more neutrophils are recruited, but also monocytes are recruited, which in the perivascular space as macrophages release all those growth factors and cytokines, which then promote collateral artery growth. So, and of course, several people have been necessary to do all those work. In my lab, in particular, Manuel Lasch did the work, but he was assisted by several co-workers and collaboration partners from other labs from Germany and all over the world. Thank you very much for your attention, and was my last slide.